Good afternoon, colleagues. Shall we begin the next session of this DIRISA track in the CHPC conference? We have with us first Dr. Sumir Panji from the University of Cape Town. He is a bioinformaticist within the H3Africa BioNet Consortium, and he's responsible for the interpretation and implementation of bioinformatics solutions, using particularly new technologies such as machine learning to address diverse biological problems. He's also actively involved in providing support to the H3Africa projects and helps develop bioinformatics capacity across the African continent. Thanks. Over to you, Sumir.
Thank you. Thank you for the introduction. Just a quick disclaimer while I get my slides up is I'm actually not a professor, so please disregard that. I don't want to get in trouble with my institution, but I'm a staff scientist and I do enjoy what I do, so thank you. So without further ado, my name is Sumir Panji. I'm from the UCT Computational Biology Division. As Dr. Peter says, I work with the H3 BioNet project and I'm based in Nikki Mulder's lab. I'd just like to thank the organizers for allowing me to speak today. So some of the topics I'll cover in my presentation today include reasons for the explosion of genomic data growth. There's a definite lack of diversity within this data explosion of genomics. I'll focus a little bit about H3Africa and H3 BioNet, just to provide you some context of the kind of work we do. And mostly for this presentation, I'll be focusing a lot on the data curation and standards projects within H3 BioNet that speaks a lot to the research theme of the conference, which is preserving valuable research assets. And also specifically focus on a couple of H3 BioNet projects that are adding value to African genomics data, as well as some of the fair assessment work we've done within H3 BioNet. So biological and particularly molecular biology has undergone a technical revolution over the past couple of decades. The initial draft of the human genome sequence actually took scientists 13 years to complete and cost around $500,000 US. Today, we can generate the same sequence at 30 times coverage for $4,000 in 1 day. And this is a graph that's from the National Institutes of Human Genomic Research. And they've been looking at sequencing costs over a period of time from 2001 to 2020. And they noticed that the costs are actually outpacing Moore's law in terms of decreasing. Just a couple of caveats with this graph though. The left scale is actually in logarithmic, and so that's why it looks a bit smooth. But also, these are just the direct cost of data generation. It doesn't include the data stewardship, the data curation, the analysis, the storage, or the transfer of the data. But nevertheless, the low data costs have created an explosion in genomic data volumes. Currently, we are expecting to reach the zettabyte level in 2025. EBI, which manages one of the major repositories for genomic data, is actually predicted to reach exabyte scale volumes in 2022. And also adding new types of biological data types, such as the biomedical imaging archive, which is the small line on the small red line on the left of the graph or the right of the graph. That's just been recently added, but I mean, the data volumes from imaging data are comparable with what's being generated right now as well. So due to this decreasing cost of genomic data, a number of countries are embarking on national genomics medicine programs. And additionally, you have numerous countries that are actually trying to start their own as well. You have different research projects that are funding national cohorts, such as the All of Us project in the NIH. And these are really important because they reflect the diversity of the populations within those countries, and they have a real public health benefits for precision medicine and using genomic data within a clinical setting, especially when it comes to dosing drugs and stuff, as you'll see. So different population groups in history have gone through migrations and bottlenecks, and so they have subtle differences in terms of the genomes, which are a mosaic from both the parents, which they inherit, which again are mosaic from the parents and parents. So we do have small or single nucleotide differences that code for letters in the DNA sequence, and this can have effects on different things as well. So we call them a single nucleotide variant when we're looking at them within our sample, but if that variant reaches a certain frequency within the population, we call it a polymorphism as well. There are larger differences, such as structural variants, but I won't go into that. The main problem is that these single nucleotide variations or polymorphisms actually affect our ability to metabolize drugs, and different populations metabolize drugs at different levels as well. And this does have ramifications, as be seen with the lawsuit by the state of Hawaii against some big pharma countries for the use of Plavix, which is similar to aspirin, but it was actually quite ineffective on the population as well. In fact, in some cases, led to an adverse outcome as well. So there's a large underrepresentation of diversity within the genomics data that's been currently being generated. And I guess that's because DNA sequencing technology, as seen in the previous slides, was quite expensive when it first came out. So the early adopters tends to be the high income countries. But as costs are decreasing, it's more accessible to low and middle income countries as well. So the technologies are being there as well. And I think there's also a problem because within Africa, I mean, as living standards are improving, so is the prevalence of other non-communicable diseases like cardiovascular disease, hypertension, and stroke. But this lack of diversity does have ramifications, especially when looking towards using genomic data in a clinical setting. So a really hot topic of research right now is polygenic risk scores. A polygenic risk score just provides an idea of relative risk scores of one's predisposition to a certain complex disease. It can be useful, but in itself, it's not a diagnosis. And these polygenic risk scores are created from current genomic data. And because there's a lack of representation of diversity in those polygenic risk scores might not be applicable to other populations as well. So the graph, the tree map on the right shows the kind of population groups that are within the polygenic risk score catalog. Mostly you can see the European in the green. And where you do have some diversity, those people are actually recruited in the high income countries that they were living in rather than being recruited from the ethnic original countries. So in order to address this, the H3Africa Initiative was created. This is to ensure that African populations are not left behind when it comes to utilizing the global revolution in genomics and genomics for healthcare. And this H3Africa initiative was born about by the National Institutes of Health, the African Society for Human Genetics, and the Wellcome Trust, which are funding the projects. Currently, there are 48 projects within H3Africa, 15 collaborative multi-site centers for population genomics, a couple of LC grants, and the footprint covers around 34 countries and with $170 million being invested to date and 54,000 research participants. So the project I'm part of is H3BioNet, which is the Pan-African Bioinformatics Network to develop capacity for H3Africa. And we work in various fields that range from training, capacity development, creating computation work tools and analysis pipelines, as well as developing infrastructure. And a lot of the work we're doing, I'm going to focus on, was mostly about the data standards and curation that we've been doing for the H3Africa data and within H3BioNet itself. So H3BioNet functions as a data coordinating center. So we collect data from the H3Africa studies. The type of data we collect could be genetic variation data, it could be whole genome sequence data. It could be just targeted sequencing where you're looking at small specific regions of genes to see if they're actually different in your population group. There's a genotyping chip array, which is a good way of giving low-cost coverage for variation in different population groups. And there's a custom H3Africa chip that's been designed, and it's a project led by Professor Nikki Mulder. And other studies include microbiome sequence data as well. Most importantly is, I mean, the data itself would just be a bunch of letters or characters or strings. In order to analyze it, the phenotype data is quite important, or we can call that the metadata in this case, and that includes demographic data, anthropometric data, disease and related health phenotype data. So as part of functioning as a data coordinating center, we receive data from the H3Africa project. So my colleague Ziad Parker is the H3Africa Data Archive Manager, and he manages a lot of this. So he interacts and sets up meetings with the sites before they're ready to submit data. He goes through them in terms of what files they require, depending on what type of analysis they've done, and to ensure that the files are correct. Once that's done, then they do transfer the data to Ziad, when Ziad then does a couple of validation checks. So because the data is quite sensitive, we have a server where it's only a few people can access it. And that's where a lot of the work is done when the data is uncompressed and unencrypted. So the kind of validation Ziad does is to check if the checksums match, are the mapping files present, all the files present for each de-identified participant ID. Is there a mismatch? Do we have the phenotype data files present? Because that's quite important. And is there a data set summary or description available? Once this passes validation, this then gets mapped to EGA schemas. So with the EGA schemas, this all in XML, and this is what Ziad does. We have what you call a high level, which is the H3Africa Consortium. And this has a consortium description. It has a data and biospecimen accession number. It has a policy accession, which is a data sharing policy that was developed by Asia Africa. And it has a project accession as well. So these three elements are common to all the Asia Africa projects that submit data to us, and we submit them to EGA. Within the consortium, you have the separate projects itself, where you have the project titles and project description, which we map to the study accessions. We do require this because this provides background information on how the data is collected and what the study is about. The project study type, is it a longitudinal, is it a trio study, is it a case control? Then that gets mapped to a data set accession. We have a data set name, a data type, a description, and we also, most importantly, the technology that the data was generated using. Then at the individual level, you have the participant IDs. These are usually control restricted access, so you do need to apply for access for this. You have the samples, then you have the runs itself, which is the experimental accession numbers, and you have the analysis, which are actually the files themselves. They could be the raw sequence files, such as the BAM or the CRAM files. And the graphic on the right just shows you that the data Ziad takes and maps into his EGA XML schemas is what he submits to EGA as well. So one of the big things that H3Bionet has been working on has been phenotype harmonization. So in terms of H3Africa data, we work with the phenotype harmonization group that's developed a set of core phenotypes, which I'll go into a little bit later. Then we take these phenotypes and we map them to CRFs themselves. CRFs are case reporting forms where the data or any measurements you take when you recruit a participant are captured on. So we try and map these to phenotype and disease ontologies just to make them a lot more findable. And also if you want to use them in scale for any large scale machine learning applications. We map the ethics consent to data use ontology and the demographic data to the ethno-linguistic ontology as well. And then we also then deposit this data to EGA and the catalog. So by mapping stuff to ontologies, it does facilitate the searching, identification, and reuse of data. Okay, there we go. So in terms of phenotype harmonization, the variables that the phenotype harmonization group have decided that will be common to all Asia Africa projects are provided on the list on the left. As you can see, it's a really high level list as well. But I guess because a lot of the projects themselves also have their own specific research focuses, which I'll show you a little bit later how that differs. But some of the problems that we do face in harmonization are the project CRFs. So on your right, you can see three types of project CRFs. So you can have the same data, but it can be coded in different ways, such as gender, female, woman, or one. Or you can have the same variable, but it's collected in different ways. So for example, how much alcohol do you consume? How many units of alcohol do you consume? So is it daily units? Is it weekly units? Is it user-defined period? And these real subtleties in how this data is collected actually do affect when you try and integrate the data for harmonization. So the phenotype harmonization working group, the structure is shown on the top left. It consists of the HIV/AIDS working group, the mental health working group, CVD working group, environmental and rare disease working group. These are special interest groups that have their own focus of research. And as HUBioNet, we provide support in terms of joint harmonization projects where we do ontology mappings, we develop minimum data dictionaries and standard CRFs. So in terms of the standard CRFs, the first step to harmonize them, and this is a lot of work that's been done by my colleagues, Catherine Johnston, Nikki Tiffin, and Lyndon Zass, that are actually leading this project as well. So the graphic on the bottom left is created by Lyndon, and this is the processes that they go through in terms of harmonizing the phenotypes. They collect the case reporting forms from the various projects. They determine the harmonization feasibility. They come up with a sublist of phenotypes to be harmonized, and then they determine which projects are collecting those phenotypes, and then develop specialized algorithms in order to harmonize the data. And just to give you an overview of the data, because they've successfully done this for this cardiovascular disease working group, or CVD working group, when they had a data hackathon jamboree, I think early last year. And the table on the right actually shows you the variables that have been connected by the different Asia Africa projects that are part of the CVD group, and what needed to be harmonized as well. So now, why do we want to go through all this effort of harmonizing data? Well, most of the complex diseases, such as cardiovascular diseases, are actually polygenic in nature. And what that means is that it's a combination of lots of genes with small effect sizes and variations that altogether contribute to an observed trait, rather than what you have as a directly one gene, one trait, which is your classic Mendelian mutations or genetics. So if you look at a graph on the left, the bottom right quadrant is actually where a lot of the polygenic diseases sit. And now, in order to see if that these variants with small effects and to actually determine if they do have any effects, you actually do need to boost the power of the study. And to boost the power of the study, you need to do that by integrating more and more samples in there. But samples can only be pulled together to increase a study's power if they're comparable. So a lot of the work Linda's been doing is he's been taking the CRS from the Asia Africa projects, he's been mapping them, and then he's been seeing what's in common with them. So the plot on the right is actually a PSA plot generated by Lyndon. And it has 2 dimensions on it. And you can see that a lot of the phenotypes, the blue lines that streak out, are projects which are collecting more or less similar phenotypes, and they can be split on 2 planes as well. So those projects would be a good starting point to actually try and harmonize the data. The other bits where you see on the right of the graph itself with no blue lines, those are actually the focus specific areas that the research projects are uh, um, doing the research in. For example, it could be malaria, it could be stroke, it could be HIV status. So just by doing that, you can automatically get a good overview of what can be harmonized and what can't be harmonized as well. And this leads to the development of standards as well. So standards are quite important because what they've done is they've actually been able to take the CRFs and map them to ontologies. And with those ontologies, they can create data dictionaries in REDCap, which are CRF modules, and also XML templates, which they can then share with other groups that want to undertake their own data collection. They do map and compare the variables with international standards such as Phoenix, and then after that, they do map them to ontologies, and then they send them out for both an internal review for consistency and external review by experts to check if the domain language and questions have been captured correctly. And also, I mean, when you do creating products, you do need to build in some kind of training and guidelines for that, which they're doing. So the graphic on the middle actually shows you an example of a data dictionary codebook for the core phenotypes, which is kind of what we're using right now. And the nice thing is Catherine and Irma actually run a monthly REDCap poll that does talk a lot about the standards specifically for African researchers that are looking to implement REDCap. And this work is actually being put to good use because Lyndon, Catherine, and Nikki Tiffin are currently working on developing a COVID-19 specific module for use in South Africa, which they hope to get published as soon as we can put up on Ziva Hub and get a DOI on that. So Asia Africa data is quite tricky because you have 2 types of data. You have the actual sequence data, which are coming from the archive, and you also have biospecimen data, which are basically DNA in a vial, which can be requested for and used for any studies or analysis. So in order to harmonize this data, we've created a metadata access catalog for H3Africa Genomics data, where we ingest data from the biorepositories and from the archive. We do process them. And then the catalog itself does provide three views. You have a public access view, you have a registered access view, and a DBAC access view. So why it's useful is because the actual search interface we have for the H3Africa data is probably a lot better than you'd get at the moment on other repositories. So you can search by primary disease, disease status, ethnicity, gender, and these are all things that we've mapped to the method as mapped as variables for the metadata rather than giving individual level variables, because we do not want to have a risk of any information being leaked as well. Within the catalog, researchers can come register, they can add data sets or biospecimens to the cart. They can create a request, which then they fill in a form in the intended research use, and that gets sent to the data access and biospecimen committee. And they determine if the access actually falls within the consent of the actual study and within the actual data sharing policy for Africa. And one of the nice things that we've implemented recently have been the data use ontology within the catalog. And I'll just talk a little bit about the data use ontology. So most data sets with phenotypic information are always going to be access controlled because of the personally identifiable information. So usually a data access committee receives a request for data access. They review it in line with the consent and the data sharing policies. But sometimes, I mean, the data sharing policies and the consent can be quite opaque in terms of the language. So it's very difficult for a reviewer to decide if this application actually falls in line with what the data has been allowed for when it was collected. So and these reviewers also might have to actually go through like 500 to 1,000 plus requests for data as well, especially when new data is released. So in order to take away some of the ambiguity, there's a data use ontology that's been created by the Global Alliance for Genomic Health. And it's part of an ecosystem that aims to create automatic data matching between users and requesters. It's quite useful because it allows a data access committee member to go through the data request and check if it is actually in line with what has been consented for and release it as well. So I'm just going to, I still have about 5 minutes left, so I'm just really going to go through these briefly. So two of the projects that we've got is creating portals for African genomics data. So one of the difficulties we face is there's actually, when you're looking for African genomics data, there are a lot of resources we have to go through. There's no single entry point that we can mine. So we decided to create a portal that mines publicly available information and creates it in a catalog. This includes variant, disease, gene, and drug. So the actual protocol is we mine PharmGKB that has pharmacogenomic information, and DisGenNet, which has disease information. We get the raw data, we map them to the PubMeds, to the publications, mine the publications with text mining, and curate them manually to ensure that we do have real African data in there. And then the other annotations, such as gene and protein, we do get them from other sources and we incorporate them into this database. Similarly, for African microgenomics, the microbiome studies, there really is no portals or unified place to find the data. So we're also creating another portals data for this. And the aim is to basically have the most comprehensive statistics available on metagenomic standards, similar to the African African precision medicine portal, where we mine public data, we do a step of curation, we harmonize it, and then we map it to different entities. And that allows you different search entry points into the interface. So the African Genomic Medicine Precision Portal is led by Prof. Salma Qasim from Ancient University in Cairo, and the microbiome portal data is led by Osama from Institute Pasteur of Tunis. Another database we do have is the African Genome Variation Database, and this is a whole different ballgame on its own. It's built using OpenCGA, which allows for variant analytics and annotation. The rationale behind this is whenever you look for African data, it's really never updated. So when you calculate frequencies, it's really never the correct frequencies are calculated because it's not been updated. And two, they tend to lump African data as in one population, whereas we do know it's a heterogeneous population and subsets of populations. And these are quite important because researchers usually want to find out that how common or rare is my gene of interest or my variation of interest. It might be rare in a European population, but it might actually be very common in the African population you're studying. But you never pick that up if you don't have a place to compare that to. Some of the work that we've been doing involves COVID-19, and this is work that's been done by Emmanuel Horaya and Prof. Tulia from CRISP, UKZN. In terms of creating and annotating genomes, I mean, the sequence itself is quite important as well because a lot of that gets used for downstream analysis. So during our AGM session, the UKZN team hosted an online practical workshop on curating COVID-19 genome sequence in South Africa for submission to the GIS-8 platform for data sharing. And it was, I think it was like a 4-hour tutorial, but it was quite good because we used a lot of tools and stuff. And the screenshots just show you the various mutations, what's the correct mutation, and what's actually a wrong mutation. And it's very important to curate the sequence data at the sequence level because once you submit it, you're using this for creating phylogenetic trees. And phylogenetic trees actually use the underlying sequence to create the distances between them. And if the sequence is actually wrong or the letter is actually a mistake, then you'll get a different tree. And it also throws off your transmission dynamics. We are also implementing standards for training workshops and materials. We had a bioschema workshop early on last year. And this is work that my colleague Verena Russ is doing. And she's designing templates that actually incorporate bioschemas in them and the descriptions for the workshops as well as licensings for the workshop materials. This is quite useful because this kind of format is quite readily indexable by search engines like Google and they get found quite easily. And the next step is to also add these to repositories such as Ziva Hub. Okay, so I'm just going to quickly focus on some of the fair work that we're doing in H3BioNet. So the H3Africa data is actually being made fair by H3BioNet. If you look at all the metrics for fair on the left of the slide is what we're actually doing today. We actually hit that for the H3Africa data. And a really nice tool, which I'm sure everyone is familiar about, is the Fair Assessment tool, which is hosted by the Australia Data Commons as well. It's quite a nice online widget. So in comes to assessing fair within H3BioNet, we have quite a few products. We have training materials, software tools, bioinformatic tools, CRFs. So we kind of figured that one form and one rubric would not be enough. So we used the Fair Shaped rubric that has different questions for different resources, created a form, and we got members to fill this in. This is not a comprehensive process. It was just to raise awareness amongst H3BioNet members that when you're creating your materials, try and create them with fair in plan. And the results, we got a bit of a mixed bag. There's some more work we can do, especially for the tools and workflows, which really interests me, is we can add EDM ontologies or tag our workflow Git pages with EDM ontologies to make them more findable. We do need to register all our tools in BioTool registry by Alexa as well. So there are little things like that. And what this is leading to is for us to create a form which you can use as a checklist to send out to consortium members to make sure the tools are there. And in conclusion, I'd just like to say the graphic on the left shows the growth of data, and this is probably outdated because this was 2015. So I think, like a lot of fields, genomics, the limiting factor for genomics now is not the data generation or the cost of generating the data. And because of that, the data is becoming increasingly multidimensional, especially when you look at national cohorts. Our biggest bottleneck is data analysis and interpretation. So we'd like to apply automation to this and stuff like machine learning. But in order to do machine learning, you need really well-labeled data, and you need lots of it that's required. So a lot of the work H3BioNet has been doing has been adding value to this data in terms of curating the data and good data stewardship practices, because this is essential for the data to be reused, and that's kind of what we're doing in H3BioNet. In conclusion, I'd just like to take the H3BioNet consortium members and everyone that's contributed to all the projects that I've briefly touched about. I'd like to thank the sponsors for the conference and the organizers for inviting me. Thank you.
Thank you, Samir, for a very interesting presentation. I must say you guys are always doing such interesting work. So I have asked the participants to share their questions with us in the chat box. But in the meantime, I know we did some investigation of the work that you're doing for the ELIFO RDM project, and we were so impressed by the way you are mapping your metadata to the EGA XML schema. And the problem that we were trying to resolve was the interoperability between the EGA archive and our local repositories, because we don't mind where you post the data as long as we have a record of where it is. So if you don't use a DOI, and it sounds like the EGI offers you one of their own identifiers, if you don't use a DOI, how do you actually cite the data set that you are using or you're referencing in a paper?
Okay. So in genomics data, so each data set will have an accession. That accession itself is meant to serve as a permanent identifier for that data set within that system. It's not like your typical DOI where you have the DOI dot prefix, but that accession is meant to be persistent. And
So it's an EGA accession number, basically.
Yes, it's a persistent identifier.
Okay, so we could
Yes.
map to that. That's all. That's fine.
Yes.
So there's another question here in the chat. What storage volumes are used by the system?
If we're talking about the archive, we are looking at probably close to 200 terabytes, if I'm not mistaken. I think we've got like close to 108, well, we've got close to 80 terabytes of data now, but we're looking at around 200 terabytes. And this is also because, don't forget, when we do the analysis, okay, so two types of storage. So for the archive itself, when you unencrypt the data, you do need a lot of storage because that is a lot of processing power.
Yeah.
And if the files are in compressed format, you need to uncompress them as well. So that does then inflate the storage by like 10 times. When you do bioinformatics analysis, the intermediate files also inflates the storage by a factor of 10 to 20. So depending on your initial data volume, try and factor in 10 to 20 times more than that for the actual processing.
Yeah, it's very impressive. I don't see any more questions, so it just remains me to thank you again, Samir, for a very, very interesting presentation. Thank you very much for being with us today. If you'll stop your video, I'd like to introduce Isak van der Walt, who is our next speaker. Isak, if you'll start your video just for the introduction, because I believe you're going to do a pre-recorded presentation. Isak is the manager of digital scholarship and innovation at the University of Pretoria. He manages the work of the Digital Scholarship Services Unit and supports faculty and students in the use of digital tools and methods in research and teaching. Isak will be speaking to us today on the topic of digital literacy frameworks and the underlying data principles thereof. Over to you, Isak.
Hi, everyone. My name is Isak van der Walt, and I am the Digital Scholarship and Innovation Manager for the Department of Library Services at the University of Pretoria. Welcome to my session. My session is about digital literacy frameworks and the underlying data principles. In this presentation, I'm just going to briefly go through what digital literacy is, why it's important, and what frameworks exist out there, and what are we planning to do at the University of Pretoria, digital literacy assessments, and then also talk about where the data is in all of this. I mean, that's the title of the presentation. And then what is the next steps, specifically for the University of Pretoria. Right, so if we look at digital literacy, the first definition of it came about the late 1990s. So that is, I think it was around 1997, where the definition stated broadly that it's the ability to understand and use information in multiple formats from a wide variety of sources when it is presented via computers, which is, I think, obviously a good definition, and it had to take into account that these are connected computers. This was sort of the internet era. Right, but the most recent one is the ability to use information communication technologies to find, evaluate, create, and communicate information requiring both cognitive and technical skills. Right, so digital literacies is not the replacement of literacies. The other literacies, information literacy, media literacy, data literacy. So it exists within this broad spectrum of multiliteracies, if I can state it like that. So, but it's focused primarily on how to, or the skills that you need to conduct yourself in a digital world and digital era. The same as, if you had to take into account prior to it, was just, information literacies, and they're still all relevant. All right, so with digital literacy, the University of the Netherlands identified 12 competence areas for digital literacy, and they published an article about it. So when I look at these 12 competence areas, the first one is general knowledge and functional skills, use in everyday life, specialized and advanced competence for work and creative expression, technology-mediated communication and collaboration, information processing and management, privacy and security, legal and ethical aspects, balanced attitude towards technology, understanding and awareness of the role of ICTs in society, learning about and with digital technologies, informed decisions on appropriate digital technologies, and then seamless use demonstrating self-efficacy. Right, so I have to connect digital literacies as well within my role and where we come from as an academic and research library. Right, so when I look at digital literacies here and the definition, I move quickly just to the definition of digital scholarship, right? So two of the definitions that I use often, I want you to just look at the definition based on the previous slides, where digital scholarship is the use of digital evidence, methods of inquiry, research, publication, and preservation to achieve scholarly and research goals. Right, and the other one that I also like basically says that it's the extensive use of one or more of the new possibilities for teaching and research opened up by the unique affordances of digital media. All right, so you can see how they are closely related and how you would need to have the digital literacy skills to be a digital scholar. Right, now this presentation is not about digital scholarship, but I just briefly want to mention some of the activities of digital scholarship. Right, one example would be data visualization, computational text analysis, the creation of digital scholarship software. So think about programming, digital collections, 3D models. Right, so if you think about these activities being a digital scholar, you would need to have the digital literacy foundations to be able to work with these the different activities, and you need to sort of be able to conduct yourself in a specific manner. All right, so digital literacy is important because as digital technologies continue to influence teaching, learning, and research, colleges and universities are developing and expanding digital literacy programs to ensure that students and educators with researchers can not only use digital tools, but also critically consume and create a variety of content. All right, so if you revert back to information literacies, the same would apply within the context where information literacy started off. Right, so you would still need to be able to have a certain set of information literacy skills to be able to write a research paper. Now, you still need those skills, but you need the additional set of skills to do this in a digital environment. Right, so the next few slides are examples of digital literacy frameworks. Now, I want you to just observe, as I go through them, the similarities between the different literacies. So the first one is the Digital Literacy Framework for Virginia Tech University. Right, now if you look at this specific one, they basically have 5 broad categories, which they classified as curiosity, reflection, equal and social justice, creativity, and participation. And then within that is identity, well-being, discovery, evaluation, ethics, creator and scholarship, communication, collaboration, and curation. Another interesting one is Digital Literacy Framework has been developed by Doug Belshaw, which has 8 elements of the digital literacies. Now, when you look at these literacies, you notice that it's obviously not, it doesn't give a lot of clarity with regards to what goes into them, because they also differ in the sense that, or they're adaptable. So when I look at cultural as an example, one of the elements, there can be a whole range of training modules or specific curriculum that will address that, and won't be the same at each institution or each country, as it differs based on societal differences, right? They have cultural, creative, constructive, communicative, confident, cognitive, critical, and civic, right? And then obviously within each one of those elements, they have different components that will need to be, needs to receive training. Another one is from the University of British Columbia, and theirs is split into 6 elements, with the first one being research and information literacy, critical thinking, problem solving and decision making, creativity and innovation, digital citizenship, communication and collaboration again, technology, operations and concepts. Now, you can again start to see how there are similarities between the different frameworks. Communication and collaboration is a very common theme throughout. Digital citizenship is also a common theme throughout. Critical thinking, problem solving, again, also throughout. Right, let's jump over to another one that is a framework for higher education. So this is the Developing Digital Literacies framework from GISC. Right, now if you look at this one, they include to highlight that the literacies do not live in isolation. They include media literacy and information literacy as part of the elements of the digital literacies. And in addition to those literacies, they have, again, communication and collaboration, career and identity management, ICT literacy, learning skills, digital scholarship, which makes up in total the 7 elements. And again, within each one of those, they have their separate components. Now, having looked at the majority of them, and also an additional framework that I found from France, and I'll explain why I looked at that one specifically as well, the following were identified as common themes. So you look at information and data, how it's being created, where it resides, how you process it, how you interact with it. Communication and collaboration, as I mentioned, very common theme throughout. Content creation, privacy, security, protection and ethics, and then your digital environment. All right, so if I look at information and data as an element of a framework, the first bit breaks down into how do I conduct research? How is it being done? Now, the same would apply in the normal context if I look at information literacy. How do I conduct research? But the only difference is now, how do I do it in a digital world? What are the tools and what are the skills required to do research in the digital world. Again, additional as well, and this is a recurring theme throughout, is managing data. Now, from a library's perspective, we've all been busy with research data management for quite some time now. But what we're seeing in terms of developments and trends at academic libraries is there's an evolution and a maturity of data management services. Right, so initially it was you need to save your data there, you need to name your data that's your file naming convention. But now it's becoming a little bit more refined, where we look into data curation, metadata administration, we have data archivists, etc. Right, so how do you manage your data when you're doing your scholarly activities? And then also, how do you process data? When I look at processing data, it takes into account my ability to work with different types of data. Can I, as an example, visualize my data? Can I run sequences on my data, my analytical skills with data? So you would need to have the skills for that. Now, we shouldn't make the assumption that no one knows how to do this, but we also shouldn't make the assumption that everybody knows how to do this, which is why we'll get to assessment a bit later. Right, so I don't have courses right there at the moment that I'm going to show you, because this will differ from institution to institution and also from individual to individual. The next element there is communication and collaboration. How do you interact in this digital world? I think we all can agree that we've been sort of thrown off guard by Zoom and Google Meet and all these things. But how do you interact in that way with the research community? And how do you interact with people in general on different platforms? I'm talking about live video interaction versus, blogs and posts. So how do you interact with the community and how do you engage? Where do you publish? How do you share your data? Which are the journals that you need to publish in? Again, it's the same with information literacy, but there might be different elements, because now we're looking at data publishing. We're looking at platforms that you can share your data on, outside of the normal journal publications, LinkedIn, Facebook, Twitter, you name it. How do you collaborate with people, and how do you fit into that digital world? Right, so how do you conduct yourself? How do you manage your identity in this digital world? I mean, we all have different identities. We have one from the social aspect, and then we now have one from, that relates to our professional profile. Right. Next element there is content creation. Now, this is where digital scholarship also plays a very important role. Now, it seems very fundamental, the first one, to create a text document, right? But we need to take into account there are different levels of creating content. So to create content in a text document, it's not just in Word, it might be in Google Docs as an example, where you can share, you can get additional input, you can work live on it, you can work multiple people on it. Then creating multimedia documents. Having to create this narrated PowerPoint might seem pretty straightforward to most, but some people might not know how to do this, and this is what you need to be able to work in this world. Document adaptation, how do you manipulate, how do you change documents, how do you change file formats, how do you just work with the content, right? Can you take different forms of content and put it into a new type of document? And then a big thing as well is programming. When you look at the increased uptake in different disciplines, from the uptake on Python and R, these programming languages that used to natively just belong into the computer science and engineering domains, now it's across discipline, and to some extent it exists in libraries as well. Other element is privacy, security, protection, and ethics. Quite a large element there. But how do you protect yourself as an individual? how do you protect your data, how do you ensure your well-being, and what are the information and data ethics that relates to what you are doing and how you are doing it. And then lastly, the digital environment relates to how do you build your environment that you work in. So you can ask any well-versed digital scholar what they use for their environment, and they will list a whole range of tools that they need to function in this environment. They will have something to create their slides, to visualize the data, to record, to basically their own virtual office, if I can put it that bluntly. And then also the ability to troubleshoot their environment. Can they resolve their own issues? Can they figure out what doesn't work and what needs to be done? All right, so these are the elements that we are looking at the University of Pretoria. Now, we need to be able to assess all of this first. All right, so why we would assess this is to determine the level of digital literacy internally to the library, because we need to determine if we can and how we are going to provide support to our clients. Right? The other one is to determine the digital literacy level of our external clients, which is the students and staff. We then need to be able to develop an appropriate curriculum, internal and external. And we need to be able to identify problematic areas and themes. Are people struggling with information and data, or are they struggling with privacy and ethics? So that's part of the reason why we have this assessment tool. And the assessment is practical, and it adapts based on your performance. So it will ask you to do specific things with files and data. And it starts off with a very easy aspect or question. And if you answer correctly, the difficulty of the questions increase to determine your level of digital literacy. So we are currently still in testing phase with this, but I would quickly just like to show you where and what this looks like. Right, so here are some of the questions that we have in this assessment tool. So this one asks an example: where was this following picture taken? Right. So those of you who are quite familiar with reverse image search will know I can save this image, upload it to Google as an example, and search where this picture resides. And then I will find where this exact picture was taken when it was published on the web. Now this was in Hazelwood in Victoria. This one asks what year this leopard is born. Right, again I have to reverse image search to find where this picture resides, and on that website is a video, and in the first 10 seconds it tells you what year this leopard was born. Now you might ask why, because I'm testing your ability to search and discover information. Another example here is what is the focal length of this following image, right? If you are aware, you can go into the metadata properties of this image and it will show you what the focal length was of this image. So I'm testing your ability to work with metadata, or seeing if you are familiar with metadata and going to go up to that level. Now, there are about 60 questions in the assessment, right? And it tests various elements. So I'm not going to go into that. But ultimately what I get back is I get how long it takes a person to do specific elements, all right, and whether they had it right or wrong. So some might even have all the questions right within an element, but they took way above average to be able to do that assessment. All right, so assessment with the digital scholarship unit, that is my unit, to determine what is a good level. I hope they're good, because they should actually have a very high level of digital literacy. Then we need to complete the assessment with the whole library to determine where we are, and then identify from there our areas of expertise and concern, and then based an internal curriculum based on the outcome of that assessment to ensure that we have a specific level of digital literacy. Right, once we are done with that and we're confident that we can provide a specific service to the overall community, we can provide access to students and staff, and they can also then determine on what they get. The next point there is it will provide them with insights in terms of what recommended courses are available for them and where they should give attention to. Right, so we don't have too much time, but throughout the presentation, I hope you were able to see that there's quite a lot of data and information at play when someone does a digital scholarship activity and how they conduct themselves. So there is not one element that should receive specific attention, but rather a whole encompassing set of data principles, like what do you need to ensure that your data is safe? What type of data are you creating? Can your data be preserved for the long term? Who can see your data? How do you share your data? Where does your data reside? How is it being stored? Now, I can't just fix this to one element within digital literacy, because this happens when a person starts writing his research proposal on a specific platform. The data principles are still relevant when that person is gathering his information. The same applies when he's busy analyzing his data, when he creates his findings, when he writes his research findings, when he publishes it, when he shares this, and to whom he shares this. Right, so we need to be mindful of all the data points that are being created. We often tend to think that just by doing a research project, the outcome is a person's final data set and the article that they're publishing. It's actually not. Data's being generated from the get-go all the way through up to the end. And the longevity of that data does not stop at a specific. So ultimately, with digital literacy, digital scholarship, we need to keep in mind that we need to have the necessary skills to conduct ourselves in this new environment. Some of us might feel that we are. I think that's the reason why we also want to do this assessment, is you might not know everything. So this is not to point someone out. This is for us to become a better equipped library, to become better equipped researchers and students ultimately at the end. So with that, I'm going to allow for questions. Thank you so much.
Sorry, thanks, Isak. You always provide a very interesting insight, I must say. Thank you very much. We have a question in the chat for you. I don't know if you can see it. Nicholas is asking, how are you working with the terminological, conceptual, and practical divides between open science and open or digital scholarship?
Right. Thank you, El. Thank you, Nicholas, for the question. It is actually quite a challenge, I must admit, because I personally feel that I think both open science and open digital scholarship operate in silos. So I think we tend to collaborate quite a lot with the Open Scholarship Office in the library specific. But I think more work needs to be done in terms of advocacy and then also probably larger working teams at the university. Now, this has not been established with the University of Pretoria, but that would be a workaround for us to make sure that we're not just steering into our own direction and have our own conceptual framework while it exists somewhere else at the university, or with, let's say as an example, they might call it something else within the open science framework or different departments. But I think probably more advocacy and probably more engagement with different faculties and supporting units is probably my answer.
Thank you very much. Yeah, I'm not alone with those challenges. So I had a similar kind of idea going through my mind, that the work you're doing is so valuable and it really needs to be applied across the university more generally. So I don't know, you may have had some experience, as we have, with the need to educate students how to conduct themselves, as you say, how to conduct yourself in the digital era. And the issue that we've been trying to address is the use of abusive language in social media and the risk that that poses to the institution. I mean, this is not a library responsibility, but it sounds like you probably be the best people to address an issue like that. Have you come across a similar kind of scenario?
We haven't. Well, I've come across examples of use like that, in terms of communication. But what we intend to do is not to become the main experts, but also a gateway to stakeholders on campus that can provide us with more insight. So we would approach someone that is a specialist in data ethics that can assist us in terms of either presenting a workshop or if it needs to be related to security, we'd rather get someone in that's sort of an expert or in the field of information security or data security. But yeah, we've come across that in terms of examples, but as a library, we can't take the full responsibility to guide that. We can play a supporting role.
Great, thanks very much. If there are any other questions that I perhaps can't see, perhaps just unmute yourself, any of the participants, if you'd like to just unmute yourself and share your question. No, I don't think so. Right, thanks. Well, then it remains for me just to thank you again, Isak, for a very interesting presentation, and then also to close the session, and hope you all have a quick break. We'll see you back here at 3:15 for the next session around the CoData Summer Schools. Thank you. Bye.